on? Oh, there we go. So we're up here near the top. So we have a few more big groups to go, but we've got to finish this one off. This was a very big group, remember, with all monophyletic. So we believe that they all, that we have everybody in the group, and we believe that we have at least all the ones we know about, and we have their common ancestor. So we've got a pretty good idea of their lineage, in spite of the fact at the very beginning with the dashed lines, there's a little bit of uncertainty, which you can probably imagine as we work our way through the protozoans. All right, these were the groups, remember, that were very much involved in secondary endosymbiosis, where a dinoflagellate, for example, would have engulfed something that had engulfed something. So it was engulfing and then being engulfed. And I think we left off with the Tom, so we'll get back into the golden algae. Interesting group of from the Chromia alveolata. The golden algae get their name obviously from their name. You can even see in the picture here, Dinobryon is a golden algae. The color comes from the predominant pigment in terms of these are photosynthetic organisms. The predominant pigment that they use for photosynthesis. If you remember from Bio 1, you can photosynthesize. There's chlorophyll A, chlorophyll B. And there's one called carotenoids, which is common found in carrots, giving them their orange color. So you can see a lighter version of carotenoids would look maybe golden in color. There's a few other photosynthetic pigments, but those are the three big ones anyway. Most of the time these guys live unicellularly, but there are some colonial versions like Dinobryon. Take a look at that picture. That's really kind of neat. There's the flagellum. So I know we talked about cilia being used in terms of filter feeders, but we're going to see that in some cases you can use a flagellum. That you would think, well, the cilia work by having a lot of them moving that water around. There's only one flagellum, but oftentimes what happens, you can see in this picture, on either side of the flagellum, those curved lines in the light blue, what that is that's a collar that's all around the flagellum. So if you think about that flagellum is wiggling back and forth inside of a confined space, you can create a vortex, kind of the water going down your sink, where water materials is brought in, and obviously food, right? Very cool. And if you're all living together, like Dinobryon in a colonial fashion, then you know what you can do is really maximize all of those flagella moving within collar cells, is what those things are called. It's like a collar, like you wear on a shirt. We've talked about this a couple of times before, the idea that many critters, what was the thing I was trying to say? When the going gets rough, they get going. So they form, Dinobryon and golden algae are known for forming cysts when the environment deteriorates. Remember what a cyst does? It's one of those things where if you can't adapt to the environment, what you might do is go into this really metabolically inactive but resistant sort of state, waiting for the environment sometime in the future to pick up, to be better. Alternation of generation. Yeah, that's something important. Let's talk about that. We're going to see this a couple of times, actually more than a couple of times. What we mean by alternation of generation, it's very simply understood, okay? When you and I undergo reproduction, we produce a single cell, the gamete, the sperm or the egg, and it's a singular cell. The egg is one cell, the sperm is one cell, they come together, and the chromosomes mix, and all that stuff you learned about in Bio 1. Well, there are some critters out there, and brown algae are one of them, laminaria, and many others, actually, as we start to get into this, especially in plants, who engage in this kind of reproduction known as alternation of generations. Now, what that means is there are two generations. Now, what we're referring to generations, the somatic cells in your body, the everyday run-of-the-mill cells, liver cells, you represent a generation of those. Oh, and then your gametes also represent a generation. So the generation that is all those other cells that aren't your gametes, that's a multicellular generation, right? You're multicellular, about 1 trillion cells, okay? But our gametes are single-celled. What many plants and protozoans do is they also have a multicellular haploid stage, right? So it's kind of like a multicellular sperm and egg, although it's not exactly that way, but in terms of the generation. So where is the blue, right? The blue is haploid in this picture. Look down in the lower right, and in this particular case, there are even a male and a female version of these haploid. But notice we've gone through meiosis up there in the upper right there in part of this, about 2 o'clock on the circle. We've gone through meiosis. So we've produced haploid cells. Meiosis always produces haploid cells. It's a given, no matter where. So those cells are now haploid, those things called zoospores. A spore, if it has a flagella, it can move. We call it a zoospore. ZOO is a root word in science. It means animal. That's where you go to see an animal at a zoo, right? So zoospores, they're not animals, of course, but they move, and that's one of the big things we always talk about with animals. Anyway, during meiosis, you produce these zoospores, and there may be one type of zoospore or one type of spore or two types, but regardless, eventually you give rise to a gametophyte. Now, those gametophytes are haploid, but notice they're multicellular. And you give rise to a male gametophyte, you give rise to a female gametophyte, and those might be separate plants or separate things, in this case separate protozoans, multicellular protozoans, or the male and female gametophytes might be on the same singular plant, right? It varies. Okay, more on that later on. All right, what do gametophytes do? Gametophytes undergo mitosis, undergo mitosis and produce lots and lots and lots and lots of gametes. The male gametophyte or the male part produces lots of sperm. The female gametophyte or the female part produces lots of eggs. It's all about the numbers here, you see. A gametophyte, being multicellular, means you can really ramp up the number of gametes you can make, right? Much different than in our cases where we only produce a single-celled gamete. And we generally don't produce, we produce a lot of sperm, but certainly females, among mammals, they don't produce a lot of eggs, right? But remember, the delivery system is a little bit different and more or less ups your chances if you engage in internal fertilization, if you think about mammals and such, than if you're just basically shedding zoospores out into the environment and hopefully find each other, which is what a lot of these plants do. All right, and then we switch back over once these sperm and egg come together, these gametes come together in fertilization. There's your familiar zygote. It's obviously diploid. And you notice it's actually growing attached to this haploid gametophyte, this female gametophyte. So the female gametophyte actually in some cases provides a little protection, a little nutrients, if you will, for the zygote, which is very interesting because if you think about it in animals, it's kind of what we look at when we see mammals, right? This idea of protecting your young. All right, and of course what happens here from the zygote is we get a multicellular diploid generation, just like you and I, all right? So see, the difference is the multicellularity of the gamete-producing structures. There's the spores, which we don't do, that mix in between, and then there's the multicellular gamete-producing structure, which produces lots of gametes. A couple of really simple terms in the upper left. If the generations look alike, if the gametophytes and the sporophytes, okay, sporophytes have spores on them. Sporophytes are always diploid. Don't ever forget that. That word we're going to see a bunch throughout the semester as we finish off, especially when we get into plants. Sporophytes, phyte is a structure, a thing. Sporophytes make spores, all right, and they make those in sporangia. And they make those spores by meiosis. So we're back in the cycle, right? But the sporophytes are always diploid. Gametophytes are always haploid. Gametophytes can only undergo mitosis, because if a gametophyte underwent meiosis, it would be half an N. Can't have that, right? Can't do that. All right, so we will come back to this a bunch of times, but I would spend some time making sure that I'm very familiar with all this terminology. Sporophyte, gametophyte, spores, and the whole process of alternation of generation. Oh God, these are so weird. The oomycetes. Oomycetes are what we call water molds, but it's not a fungus again, right? We've talked about this idea of things looking like something. So I put molds in quotes to remind you of that. There's a water mold in the upper right. Fish, if they die in an aquarium, you leave them there for a while. I know this from personal experience. Unfortunately for a fish, they'll get this white fuzz around them. That's a water mold. It's also what caused the potato blight. Those of you of Irish descent may have your ancestors. Okay, let's look at this next group down here in the red, right in the middle of the page, the Rhizaria. A lot of these are amoebas, but let's be cautious with that word. Being an amoeba doesn't mean that you're monophyletic. So there's lots of things that we call amoebas. So again, it would be like thinking that all things with the word or with the name amoeba in them are related is like thinking that all flying things are related, okay? We've made that analogy a couple of times. The thing about the Rhizaria that right away kind of distinguish them as not your typical amoeba is the fact that the pseudopodia, those false feet that amoeba make, remember there's three ways that protozoans move. Their cilia, their flagella, and their pseudopods. If you look in that picture of that one down below, this rhizarian amoeba, those are pseudopods, but they're very thin. That's a characteristic of members of this group. Rhizarians make very thin pseudopods. And we're going to look at three of them. They're cercozoans, forams, which you're familiar with, and then something called the radiolarians. The cercozoans are an extremely diverse group found all over the place. Soil, marine, freshwater, move around with pseudopods, or just because you're in a group and you're known by having pseudopods doesn't mean you don't have flagella as well. Remember, there's a host of characteristics that put organisms in a group. The coolest thing I think about cercozoans, though, look in the picture, Polynella chromatophora. It gets its name because inside is a chromatophore, that green thing that they're pointing to. Chroma in science means color. So you can see that this is obviously, something, a phore. We talked about the other day in Rhizopus that a phore means the stalk that holds it up. Oh, okay, so this is a thin sort of structure, this green thing. Anyway, what is so cool about a chromatophore is their evidence for endosymbiosis, because a chromatophore is surrounded by a membrane of peptidoglycan. And you don't find peptidoglycan membranes in eukaryotes, do you? They're only in prokaryotes. Right? That's the peptidoglycan layer that makes the difference between gram-positives and gram-negatives. See what detective work does for you? Somebody figured out that was a peptidoglycan membrane around this protozoan, this eukaryote, and went, whoa, that had to have been because something engulfed some sort of prokaryote. Forams, the stuff that look like broken up shells. There are thread-like pseudopods. Now, they use those pseudopods for capturing food, okay, because you could see how you could get tangled up in those woods, kind of like jellyfish, right? Although they don't have stinging cells. For swimming, for moving about a little bit, you're pretty plankton-like in your lifestyle. You're going to float, but you might be able to move around in a small area. So where something goes from being plankton to being nectonic, remember the things that can swim like fish, is sometimes a gray area, right? It depends on sort of distance. These thin podia too, having so many like that, also help in terms of floating. Organisms, small little organisms, have lots of projections. It's kind of like if you're trying to float in the ocean and you put your arms and your legs out, you get a little more surface area that way. Unicellular heterotrophic, so these are not part of that photosynthetic group of floating things, of plankton. Marine, I don't think there are freshwater foraminiferans. As we talked, calcium carbonate is their test. There's the White Cliffs of Dover, by the way. The White Cliffs of Dover on the southeast coast of England is basically the remnants, oh my gosh, just think about it, the remnants of millions of years of foraminiferans dying, falling to the bottom of the ocean, the tests being left behind, the calcium carbonate, and building up. Now these things, remember, they're microscopic. Look at the picture in the upper right. That's a quarter of a millimeter. These are small. You saw them under the microscope. Just imagine how many forams have died to make that White Cliffs of Dover. Now, the way it got above land, or above water, is a tectonic plates. The plates pushed together, and this would have been uplifted, and that's why it looks the way they did, or the way it does. Foraminiferans, cool group. Don't see them around schools anymore. The radiolarians, again, thin pseudopods. A skeleton made of silica. There's a neat little picture of the. I should change that word to test, actually. Skeleton test. It's more properly a test. Skeleton's not a good word. I'll have to remember to switch that. But you guys know the difference, I'm sure. Pseudopods sticking out, swimming, feeding, floating, all that cool stuff. Radiolarians tend to look more roundish than the other ones, and I think that's where they got their name radial or radio. I don't think they transmit a radio station, so I don't think it's that. Out of the rhizarians now, we go to the Archaeoblastida. This is another monophyletic group, all from an ancestor that engulfs some cyanobacteria. But that's all cool. These are the red algae, which we'll talk about. The green algae, which again are kind of neat because they demonstrate the divisions between the latter that I've talked about with Aristotle, right? Where this idea that, chlorophytes are green algae, charophytes, as you see in the word there, those are both green algae. Those are both on the line to land plants, which aren't protozoans at all, right? Very, very odd, like we talked about. But chlorophytes, I've heard referred to as being more a protozoan-like green algae, and charophytes being more of a plant-like green algae. So, see what I mean? Nothing fits into a category. It just demonstrates this idea that organisms represent all these variations in between. So species can mutate into another. That was very central to Darwin's theory, remember. Let's look at the red algae first. Red algae do photosynthesize, as we learned on the lab practical, but I did remove. There's some red algae on the right. This is the stuff that wraps around the sushi, nori. So that's that. It doesn't look red once it's dried up. It has more of a greenish color to it. Multicellular protozoans, very common in coastal oceans, marine environments, very common in deeper water. And that's the neat thing. All right. Red algae photosynthesize, so they have chlorophyll. It's not that they don't have chlorophyll. They do. But they also have another pigment in them. It's something called a phycoerythrin. Erythrin means red. Erythrocytes, for example, are red blood cells. Phycoerythrin has a red color. And because it's kind of like the gram-positive and gram-negative stains, because there's so much phycoerythrin in a red algae, which we'll get to in just a second why there is that way, it makes them look red. So the green is there, the chlorophyll's there. Maybe that's what happens with nori. Maybe when it dries out, the red pigment disintegrates and the green is left behind. Huh. Who knows? Anyway, what's so interesting about this phycoerythrin, right? It's an accessory pigment. What we mean by that is it helps absorb light that chlorophyll cannot. Okay. Chlorophyll, if you remember, does not absorb green light. It absorbs blues and absorbs reds. Now, the blues, you didn't really talk about, I'm sure, in photosynthesis too much because most of the activity was up in the red range. Now, take a look at that picture right there, or that chart that I've got. When you go into the ocean, as you go down in depth, light hitting the ocean becomes attenuated, dampened, filtered out, if you will, right? Those lights, those colors of light that have the short wavelengths, but lots of energy, if you remember, like in the purples and the blues, penetrate further. They have more energy. They hit the water harder, if that's the way to describe it. The ones that are very long wavelengths, like the reds and the oranges, they don't have as much energy, so they get attenuated or filtered out pretty quick within the first few dozen meters, if you look at that. I remember scuba diving one time in the Bahamas, just down at 20 feet. I was doing a research project in college, and we were setting up some grid lines with rope and taking my dive knife and cutting rope and tying off pieces, and I accidentally sliced myself. No big deal with a knife. We've done that a million times. What was so cool is even at that depth, my blood looked very green. It didn't look red. Remember, things are the color they are because of the wavelength of light they reflect. In other words, they don't absorb. Well, when you go down in depths in the ocean, the red light gets filtered out so quick, even for 20 feet like where I was, there is no red light to be reflected. So a red algae at the depths that they can occur down to several hundred meters, what was I going to say? Wouldn't look red. Well, you wouldn't be able to see it because it'd be dark, very dark, or close to almost very dark. But if you lit it up like they do in the picture, then it would look red. You see, because the light would be, the flashlight would be putting red light onto it. But there's no red light down there. So the point I'm trying to make with all this roundabout way that I'm describing this is red algae can live very deep because of the phycoerythrin, which actually can absorb more blues and more purples and more of the light that penetrates to that end and use it for photosynthesis. See, that's kind of neat. You can do that sometimes. So what a phycoerythrin does is absorbs the light or the light energy and then passes it to chlorophyll. And then chlorophyll does what it does. Chlorophyll doesn't care where it got that energy from. It could absorb it from the sunlight itself or get it from another pigment like this. That's why it's always kind of people say, oh, red algae, they photosynthesize, but they're not green. And I learned in biology that they have to be green. Well, you don't, as Kermit says, it's good to be green, but you don't have to be. All right, and green light too, and which is weird, right? Because remember, plants look green because they do a lot of reflecting. Usually green light is not important for plants, but this phycoerythrin actually will absorb some green. So it does a pretty good job down at these light colors that penetrate deepest. Hence, they live deepest. Now we jump between the latter here, right? Looking at the green algae, they're found all over the place. There's not a lot of marine species. There's a lot of what we call brackish species. Brackish species are brackish means that the water's not salty, but it's not fresh. Indian River, a lot of the intercoastal waterway here in Florida can be brackish. It can vary in the amount of salt. We talked about lichens. Oh, and fungi in our lab the other day. A lot of times the symbiote, the photosynthetic symbiote, is a green algae. Again, a group that's got a lot of work before we fill it in. It is a paraphyletic group, and again, that kind of reflects the idea that some of them, like the or the wait a minute, that's backwards. Caryophytes aren't the ancestors to. Yes, they are. Excuse me. I presented backwards. The chlorophytes are more protozoan-like, right? Like the unicellular Calamidomonas. That's the one on the right. What did he just say? Calamidomonas. The one on the right. Volvox, large, cooperative living, cell specialization, that kind of cell specialization, cell specialization, cells doing different work, division of labor, daughter colonies, all very kind of on the road, if you will, evolutionarily speaking. Not that we're saying that you have to be on the road, but on the road to kind of this multicellularity that we haven't really seen until we get to animals, to be honest with you. Well, tissues, right? The caryophytes, on the other hand, are more of the green algae that are more plant-like. And as I said, chara, caryophyte, CH in science is a K sound, often. Those are the ancestor to the land plants. And, again, both called green algae. But when, you call something like that green algae, you can, especially in a paraphyletic group like this is, you're talking about those critters that are kind of all called by the same sort of common thing, but are really technically kind of more on one side than the other. Aristotle. Here's some green algae from Florida, by the way. We walk along the beach, you see those green, almost looks like green cellophane wrap. That's ulva. That's a multi-celled sea lettuce. Ulva lactuca. On the right is caulerpa. Now, we have caulerpa that grows in those brackish waters I was mentioning. Very cool organism, single-celled multinucleate. And if that sounds familiar, then you're remembering our fungal chapter. And if I haven't done the fungal chapter yet, because I use this later on in semesters, then you'll hear about it when we talk about fungus. That's a neat thing to do, to be single-celled and multinucleate. There are advantages to being that way, right? We have obviously taken a different route, but caulerpa, being one of them protozoans that can kind of try different things, has gone with this route. The amoebozoa. Again, the idea of being called an amoeba, as we saw in the rhizarians, doesn't mean that you're necessarily this typical looking amoeba that we see right here. With not thin pseudopods. This time we're going to have the lobe-like ones, the big ones, right? The ones that you're familiar with. In that picture of the amoeba, I'm hoping that you recognize what a CV is. Okay, that would be a contractile vacuole, and if we were doing this live, I would then ask the question, like I did in lab, what kind of environment does this critter live in? And hopefully you would understand that it needs to get rid of excess water, and your answer would come to you very quickly. Oh, slime molds fit in this group. I forgot about these guys. Weird. Look at all these. I just went online and typed in slime mold and got some pictures. They look like little eggs. All this yellow stuff that looks like it's erupting from some tree stump. Very weird. They're not molds, by the way. They're not molds. Molds, right? Molds are fungus. Actually, the reason why they may have gotten their idea that they were molds is we call them fungus animals. Mycetozoans. There we go. A mycologist is somebody who studies fungus. And zoan is animal. The reason why, again, goes back to this idea. And when we talk about animals, we'll discuss this again in very much detail. Animals are known for from other critters is active directed movement. Yeah, I know protozoans can swim around with the cilium flagella, but for other reasons they're not animals, right? But animals are this group that we generally associate with this idea of movement. And these things can move around, but they also make spores. So you can see the fungus animals, because spores is something. That's what a lot of that stuff that you see in the picture are spores. Not related, right? Not related. Just because they're moving fungus doesn't mean that they're related. Even though I did mention that, or will mention again, that fungus are actually more closely related to animals than the other big groups, like the plants, for example, that you guys might think about. These molds, plasmodial slime molds. First of all, let me make sure that you're not you're not potentially going to be confused. Plasmodium was also the protozoan that causes malaria. Okay, so is the one that the mosquito carries around. So you're not seeing double. They're using the same word. Why? I don't know. If you look it up, there's probably some reason why. I think maybe a plasmodium refers to, as I said, these large multinucleate cells. All right. So all this stuff, and if you ever want to see really cool videos of this, go on YouTube, type in weird stuff growing in the subway. It's invariably somebody's got a picture of a slime mold, okay? And if they do time-lapse photography or time-lapse video of it, this thing spreads out. So you think it's some alien thing. And they take on all these weird colors. They're decomposers, right? So they don't have to photosynthesize. Decomposers even will consume, engulf, slime over bacteria, right? Break down other organic material. That's the feeding phase. Now, what's really bizarre about these critters is that orange stuff in the upper picture, in the upper left, is one cell. That's a large multinucleate cell. Something that fungi do, okay? But these guys do it too. That doesn't make them a fungus, right? Many different characteristics puts an organism in a group, not just one. The way they get these multinucleate cells, by the way, and I think that's probably an obvious statement underneath. They undergo mitosis in synchrony. Well, maybe that's not an obvious statement. So what I mean by that is that's how this thing grows. Everybody undergoes mitosis. Ready, go, and the thing grows. So the time's it takes a little while. It's not that fast. You don't have to worry about being engulfed by one, unless you move really slow. But the time of the sped up videography is that synchronous mitosis. Okay, now, moving around and sliming around like this with the orange thing is great, but during times when food or moisture is scarce, these guys are going to form sporangia, where they produce spores. Spores are resistant. Spores are anticipation of a changing environment, all those things that we've talked about why you make spores. So organisms, it's kind of a little confusing sometimes. Do they make spores when it's a good environment? Yeah. Do they make spores when it's a bad environment? Yeah. Right? So they make some species and sometimes make spores in a good environment because they're like, wow, it's good. Toadstools pop up from underground and have their spores because they know, wow, it's wet. Great place to have children. Okay? Sometimes it's when things are getting rough because you're going into the variation. You're like, I don't know what's going to happen, so I'm going to mix the genetics up and hope one of these things sticks. One of these things works, right? Now, I got one more bizarre mold thing. It's still a fungus. This is the cellular slime molds, right? Very interesting group because they, they seem to engage in behavior, all right? Kind of. Look in the picture in the bottom. Find number one, and we're moving around clockwise, okay? Number one, what we're looking at here is these organisms spend their lives as individuals many times. So the individual organisms act like separate little amoebas. Again, remember amoeba means you're sort of a creepy crawly kind of thing. Doesn't mean you're related, okay? They move through the soil ingesting bacteria, kind of like the plasmodium ones do. But when food is scarce, okay, when times get tough, it's good to have friends. So what they do is these individual organisms, these amoeba things, begin to form a slug when time gets rough. That's what we're calling that thing down to number one. Number one are aggregating amoebas. That lump you see, there's a bunch of them growing together. Now what you're following 1 through 6 is time-lapse, basically. So the slug begins to grow, it begins to get larger. And number 3, it kind of falls over a number 4. So there's a slug, right? Continue on with the slug. Number 5, the slug begins to stand up. All right, and at the very tip, in number 6, the slug forms a fruiting body, like a toadstool in a mushroom that we talked about. They form spores at the top, right? The whole point was to make spores. All right, now, what's so neat about this? This is a single-celled protozoan, most of the time, but during these times, these single-celled individuals come together and work to produce this slug, which stands up to make a fruiting body, to the point where different amoebas that can follow them realize that their fate is to be part of that stalk that's holding it up or part of the fruiting body. They seem to get together and they seem to somehow, chemically I would imagine, understand who does what. Very simple developmental system, right? Very simple because what it does is as biologists, we can study the way this happens. How do those aggregating amoebas know, oh, you're a stalk amoeba, you're going to be a fruity? How do they know to start off with? So that's something that we always want to know about our cells. Remember, our cells have all the chromosomes and genes of every other cell, but how do cells know what they're going to be? I'm going to be a heart cell. I'm going to be a liver cell. Now, we know a lot about that, but this is kind of interesting because we're seeing it at a very cellular yet organismal level, right? These cellular slime molds. Check out the videos online on those. There's some really cool stuff. These guys are the typical amoeba. Amoeba proteus is the amoeba that you've seen under the microscope several times. Very diverse group of things we call gymna amoebas, found all over the place. Active predators, as you can see in the lower left, that paramecium's getting a little too close. They say active, but there's a video I have on, I think I put it in you guys' stuff on Canvas, of an amoeba engulfing a paramecium, and you're always like, why doesn't the paramecium go away? We talked to parameciums. How do they know things? Well, how do they not know they're being engulfed? These amoebas, they don't do it that fast. Okay, but some are detritivores. What's a detritivore? A vore is to eat. Detritus is decaying organic material. It's like compost, maybe. If you dug down in the soil, you would get past that point deep enough where you could recognize, oh, that's a leaf, that's a stick. It would be this nutrient-rich mush. If you dug down into the bottom of like a stream or a lake, you'd find the same thing. There'd be the leaves you can identify on the top, but eventually you become this mush stuff. That's detritus. And our critters actually feed on that. Amoebas are known for that. Amoebas also feeding on your brain. All right, that's an awful transition there, but the whole idea is these organisms, of course, just are doing what they do, and this particular amoeba, has a thing for protein-rich environments. So a good place to find that is in the brain. All right, and vertebrates are one of the best places to get into it. In some invertebrates, like for example, the one on the right, Naegleri fowleri, that's the brain amoeba that we hear a lot about in Florida, and that's it's brain tissue in the purple. And the little lumps there are the amoebas and other and waste products from as its waste it's digesting through the brain. So you can see that's not a good thing because, of course, you don't want parts of your brain being eaten away by some sort of an amoeba. But like as I said, what they do is they love these protein. Your brain's a lot of protein-rich environments. Interestingly enough, the one here in Florida, the Naegleri fowleri in the picture there, it gets into your body. Okay, in Florida, the danger is when you have warm, still waters. Okay, that's the danger sign for these amoebas. You probably have heard that. The reason why is these amoebas make cysts, like lots of other amoebas do, and those cysts actually settle down in the bottom, down where all that detritus was. Well, in these warm, still waters, if you jump in and you're kicking around and splashing, or something happens and it stirs up the bottom, these cysts go up into the water. And you're jumping in and playing around, and it happens. You're jumping into the water and get water up your nose. Of course, that happens. But these amoebas get up into your nose, and then they actually move themselves along the nerve that connects your nose cells, your olfactory sense, to your brain, the olfactory nerve, right? It actually inches its way along the olfactory brain until it goes up, gets to the skull, goes through the opening, because there's a little opening where the nerve goes through, squeezes through that opening, and there it's inside your brain. So that's how it gets there. Almost always fatal, too. I think 97% fatal. So you want to stay out of warm, still waters, summertime kind of stuff, right? Or you certainly want to make sure that you don't jump into the water if you're in those kind of areas. The entamoeba histolytica down there on the right causes what we call amoebic dysentery. The histolytica, what it does, lytica, we're referring to is the fact that it destroys tissue. Histolytica means to destroy tissue. So it lyses tissues. So, and dysentery is symptomatic of dysentery would be chronic diarrhea. And we've talked about that's one of the ways to die. Chronic diarrhea, meaning that you basically dehydrate to death. Humans host about a half dozen species, which some are just kind of there and not doing much, but some, like these guys here, are pathogenic, right? Disease-causing. The last group. The last group, really, it's just going to be this one slide because there's really only a couple of things I need to say about this. Because if you look in this group, the Opisthoconta, opisto means rear, okay, is in the front and the back. Cont refers to a flagella. So this is an organism that has a rear facing flagella. Remember the Euglena, for example, right? In Euglena up in the excavata, the flagella was on the front of the organism, so it wasn't a rear. So if you're thinking about things that you might be familiar with that have rear-facing flagella, I don't have a picture of one. A sperm cell has a rear-facing flagella, okay? So in this group of the Opisthoconta, of this eukaryotic supergroup, guess who you find? You find the animals. And you find the fungi in there, too. Now, the other two, nuclarids, there's a nuclarid in that picture. There's its rear-facing flagella. The nuclarids and the choanoflagellates. Choanoflagellates, there's some pictures of choanoflagellates in the upper right there, too. They're protozoans, right? Mixed in with the fungus and the animals. All right, now, I'll say this now, but I'll come back to it when we get to animals in a few weeks. But if you look at a choanoflagellate, right, a protozoan, and then you look at these things that we call sponge choanocytes, which we'll look at them when we go through them in lecture. Sponge choanocytes are found on the inside of a sponge. A sponge is an animal, folks. And what those sponge choanocytes do is they whip that singular flagella around, just like dinobryon, just like the golden algae. They whip that around and they create a vortex in those collar cells. And they're used for filtering. Sponges are filter feeders, so are choanoflagellates. But isn't it interesting that choanoflagellates, which are protozoans, and are known to be colonial, by the way, okay, look a lot like a bunch of choanocytes in a sponge, which is a multicellular animal, right? It's a fine gradient between those two. It's a fine gradient when we think about these major groups. So, again, the big thing about this when it comes to the protozoans, as I mentioned, I'm sure more times than I can remember, is the diversity, right? The mixture, the fact that you look in this group, and not only are there just different, really bizarre kinds of protozoans, and we're not sure how they all fit together. I mean, this is a polyphyletic name, this protozoan. But in there are critters in between, like the green algae, and then there's some fungi and plants and animals thrown in there, too. So, again, you look at the animals, for example, it says that we're more closely related to a protozoan, let alone a fungus, than we are to land plants. Which, when you think about critters on the planet, it's the land plants and the animals that are two big ones, right? Speaking of protozoans on the planet, protozoans exist, what's the word I'm looking for? Engage, there we go, in all kinds of different relationships, provide all kinds of different services, if you will, for an ecosystem. Like down below, there's a lot of protozoan producers. All right, producers are organisms, again, that produce something. This is the photosynthesizers. They're producing food. Every ecosystem, right, it's got to have somebody who can do that photosynthesis trick. Maybe a plant, could be a blue-green algae. There are prokaryotic producers, right? Could be a protozoan producer. Somebody's got to be able to do that. We can't do it, us heterotrophs. Somebody's got to be able to do that. And many protozoans are very important for that. So they're protozoan producers, and then things eat them. You got herbivorous plankton that feed upon them, other consumers, a lot of filter feeders. They provide a lot of food. As a matter of fact, about a third of the world's aquatic productivity, which is the picture I got down below, comes from protozoans. Very important. About a third of the world's productivity, aquatic is a term that can refer to freshwater and or fresh and saltwater. So I'm not sure exactly, but about a third of all aquatic productivity on Earth is protozoan producers. Symbionts, the potato blight we already talked about. That's a symbiont. Symbiotic relationships don't have to be good. The potato blight is a parasitic one, so it's a plus and a minus for that poor potato sitting there. Those that are involved in wood digestion. I'll get to those zoanthella in just a minute. I just want to say that one for the last. Wood digestion, in the lower right, that's a protozoan that you're seeing there called like hypermastigote. It's found in the gut of a termite. Termites can't digest your wood by themselves. They're like the cows. But isn't that kind of interesting, huh? In a little tiny termite, there's a little tiny protozoan that's helping that termite do its thing of destroying our house. Zoanthellae. All right, corals are beautiful. Right? If you haven't been down to the Florida Keys and done any scuba diving or snorkeling around the corals, you really need to do that. It's absolutely breathtaking. And I think probably one of the most beautiful things about coral, of course, everybody thinks about is color, a wide diversity in color. By the way, the only intact, well, what am I trying to, no. The only coral reef we have in the intact United States, a continuous, so we're not talking Alaska, they don't have coral reefs, and we're not talking Hawaii, okay? But the only one that's found in the rest of the 48 United States is the Florida Keys. So, and it's being heavily impacted by all the pollution and other kinds of things, as well as tourism that's impacting it. See, corals are animals, kind of sensitive. See the little inset picture there with the yellow? Those are what we call coral polyps. And corals are related to jellyfish. Those are tentacles. And they use those tentacles to capture and sting prey and do what they do with tentacles. But we'll come back to corals and animals when we get into animals, but let's go back to the protozoans, the things we call the zooanthellae. In the picture in the upper right, the zooanthellae are the little red dots that are scattered throughout all of those coral animal cells. All right, so this is a protozoan that lives in the coral tissue. Okay, it's actually what gives the coral its color. Coral has its color, greenish or yellowish, not because corals are a jellyfish. Again, they're kind of clear, transparent or not transparent, translucent, whatever the word you want to use. You've seen a jellyfish. You look at the tentacles. It's the color, though, that comes from the zooanthellae, these symbiotic protozoans that live in there. All right, now, what do? Well, these zooanthellae photosynthesize. Guess what we got now? We got an animal using its tentacles to acquire food, but the zooanthellae, which are photosynthesizing, right, which are making food, are making more than they need. So the coral says, hey, how about you and I live together? I'll provide you a place to live. I'll provide some carbon that you need. You photosynthesize for me and make food. The extra that you don't need, I'll use for food. And they live together in this symbiotic relationship. So next time you see a beautiful coral reef, you think about it. The color comes from these little symbionts that live in there. Now, the sad part is something that we deal with down the Florida Keys and other areas of the world, especially the Great Barrier Reef in Australia. My son was down there, and he said it's not what you would think it would be. It's unfortunately is what we call coral bleaching disease, and corals become white during this color, and oftentimes they die. What corals seem to do when they're stressed, how does a coral get stressed? Well, the environment. It gets too warm, gets too cloudy, it has some pollutants in it, whatever the case may be. Corals are known to expel, kick out, get rid of their zooanthellae. And we're not really sure why they do that, because it's very often a death sentence. See, the coral has lived in the symbiotic relationship with the zooanthellae so long, it can't take care of itself, feed itself enough by itself. So sometimes the coral gets stressed and kicks out its zooanthellae, and later on it absorbs them back up and it survives. But very often, especially if whatever is stressing the coral remains around, like I said, if the water gets too cold or water gets too warm, corals like warm water, but not too warm, these organisms will expel their zooanthellae and not absorb them and actually will die off. And it's called coral bleaching. You can look it up online. It's tragic. They show some beautiful, there's one picture I remember distinctly of brain coral. And I do have these pictures when we look at the animals later on. But on the left is one that's healthy, and it's got a greenish, kind of dark greenish brown color like brain coral does. And on the right, it's completely white. Same head of coral. Same head of corals. It's horrible. Okay? We'll talk more about that when we get to the animals. So that's it for the rest of the protozoan chapter. If you have any questions, please let me know.